Assalamu alaikum and a very good morning. Today's lecture is about product analysis, and in this we will be discussing about the overall product analysis which is involved in the making of any biopharmaceutical product. A little bit about myself. As you can see, my name is Nadeem. I am a postgraduate of MPhil Biotechnology from Dow University of Health Sciences, and am currently serving as an instructor at Dow College of Biotechnology. So starting off, the main learning objectives of this lecture would be for the students to familiarize themselves with the introduction to product analysis, the basic terminology that are involved in product analysis, the potential impurities that they can encounter in biopharmaceuticals, and the removal of those impurities from the product stream. And we will be also discussing about different bioassays as well as immunoassays. So all pharmaceutical finished products, they require quality control testing so that we can make sure that the product is up to the certain specifications which are already predetermined. This is done through potency testing, which is for the efficacy of the drug or the biopharmaceutical, that it makes sure that the drug is efficacious when it is administered to the patient. And also a prominent aspect of safety testing is that we need to check any product for the potential contaminants. So what we're seeing is that whenever we're talking about the biopharmaceuticals, the range and the complexity of the testing or the quality control testing that is done, it is far more than the testing that is done for normal pharmaceuticals which are made through organic synthesis. This is because the biopharmaceuticals, as you know, they are larger in size and they are more structurally complex as compared to the smaller components which are made in traditional pharmaceuticals. And as we know that proteins are the main component of any biopharmaceutical, so also we need to make sure the production of biopharmaceuticals in cell cultures or microorganisms, that they are also free from any kind of potential contaminants. Now, quality control. So quality control testing is done in finished products to desired specifications. As we know, the quality control testing is an essential part of the pharmaceutical manufacturing process. It involves the testing product to make sure that it meets the predetermined set standards for its potency, efficacy, as well as its safety. So the potency testing, it is done for the ensuring that the drug is actually potent when it is administered inside the body. And the safety testing, it is done to ensure that the drug product is free from the contaminants, any potential contaminants, especially in the case of biopharmaceuticals. As we know, it could be faced with potential contaminants that could harm the patient or could also render the drug ineffective. As we discussed this previously, that the complexity for the biopharmaceuticals, the testing is far greater than that of the traditional biopharmaceuticals, the traditional pharmaceuticals, sorry, due to their size being much larger and the complexity as compared to the traditional low molecular mass drugs. Talking about the potential impurities of biopharmaceuticals, the most significant risk can be of microbial organisms or microorganisms The most significant risk that is associated with microorganisms is the contamination of or the establishment of a severe microbial infection such as septicemia. As you know, microorganisms can arise from various sources. This can be the raw materials, the equipment that is being used, the manufacturing environment itself can be a potential harbor of microorganisms. So it is essential to identify these contaminations so that we can prevent them further, or we can prevent the spread of infection through the biopharmaceutical product, ensuring patient safety. Similarly, viral particles, they can also cause severe infection in the patient, and viral contamination can also arise from different sources. They can be from the raw materials, they can be from the equipment used, or throughout the process. As viral contamination can be a little bit more challenging, as viruses are far more harder to detect than the traditional microorganisms or the bacteria and other things. Then there are pyrogenic substances. They can cause a fever response in the patient, or and can also lead in certain cases to the death. These can, as you can say, these are usually coming into the product from the water that is being used, or if the glassware is not washed properly or properly depyrogenized. So these are the potential sources that can introduce pyrogenic substances into the product. DNA is also another potential contaminant that can bring about the immunological response in the patient. However, the significance of DNA is still a little bit unclear, but it is also very important that we need to identify the DNA contamination so that we can prevent any potential adverse effects in the patient or the receiver of the said biopharmaceutical. Then also, when we're talking about contaminating proteins, as we know, there is also an immunological reaction or immunological risk effect involved through the introduction of contaminated proteins into the biopharmaceutical. Therefore, it is also important that we get rid of these proteins as well. Talking about protein-based contaminants, the chromatography is done to separate the proteins of interest from other contaminated proteins. Chromatography is a widely used method that is done for purifying proteins. It is based on the principle of selective absorption and elution. In this method, the protein of interest is separated from the contaminating proteins based on the physicochemical properties. Now, the choice of chromatography method depends on the specific properties of the protein of interest or the contaminants. One of the essential steps that is done in protein purification is to remove the contaminant that can interfere with the properties of the protein of interest. It can be through filtration, or it can also be done through chromatography. Now, in case we have a product that is of intracellular origins, that is, the product is being produced intracellularly, so we may require additional purification steps for such proteins which are produced in large quantities in the host cells. Now, what happens is that these proteins can be challenging to separate from the host cell proteins or other contaminants, and we may require even further additional steps which would be required to get the desired purity of the protein of interest. As we know that the origin of protein-based contaminants can be from source materials such as the raw materials of the manufacturing processes, or it could be introduced during the upstream processing, or the downstream processes. We need to make sure that not even minor amounts of proteins are entered in the product stream, because they could be potentially hazardous to the patient. The minor amount of protein that could come into the product stream could be from the product personnel, Therefore, we need to make sure that proper good manufacturing practices are being utilized in any kind of product that we are producing, in case of any biopharmaceutical product. The clinical significance of protein-based contaminants or protein-based impurities, they can relate to two key factors. One is their potential biological activity, and two is their antigenicity. Firstly, some contaminants may exhibit properties that are deleterious to either the product itself or the patient. The example could be the presence of proteases in a product, which can potentially degrade the protein of interest, therefore losing the activity or the efficacy. Similarly, it could be the presence of contaminated toxins that could harm patients. In the second part, their antigenicity or the antigenicity of the protein-based contaminants is a concern. Contaminants which are foreign to the human body can elicit an immunological response in the patient's body, which could potentially lead to an allergic reaction or immune system activation. This is relevant in the case of biopharmaceuticals, which are usually produced using recombinant DNA technology or other biological systems other than human settings, where the protein-based contamination risk is far higher. Now, the protein-based impurities are not limited to the contaminants, but can also be in the form of modified proteins of interest. These modified proteins of interest can also compromise the quality and quantity of the finished product. Therefore, we need to make sure that they are also removed. Now, one significant issue that arises with the biological inactive form of proteins is the reduction of the overall protein potency of the final product, and this reduction can lead to treatment failure or reduced efficacy. Therefore, we need to make sure that these are also removed from the final product. Also, the modified product forms may remain biologically active, but they can exhibit modified pharmacokinetic characteristics. What I'm trying to tell you is that the clearance of the body may be different. The pharmacokinetic, basically, as you can say, the clearance of those products from your body may take longer or even faster, which may also lead to different dosing requirements for the patient. Now, another potential issue with the modified product form is their immunogenicity, as in they can also elicit an immune response in the patient, which could result in the allergic reaction or any other adverse effects for the patient. Now, therefore, it is crucially essential to make sure the quality and potency of safety of the biopharmaceuticals is clearly monitored throughout the process. So we have established that we need to remove the altered product of interest from the product stream. So if we can alter the physicochemical properties of the modified protein of interest, we can remove it through chromatographic techniques. The chromatographic techniques could be such as ion chromatography, ion exchange chromatography, it could be size exclusion chromatography or hydrophobic interactive chromatography, which can be used to separate the modified product of interest from the other impurities based on the property. For example, if the modified protein of interest has a different charge than the other impurities in the product stream, we can use ion exchange chromatography to separate the product of interest from the impurities based on the charge differences. And if the modified product of interest has a different size or hydrophobicity than the impurities, then size exclusion chromatography or hydrophobic interactive chromatography can be used to separate those proteins. We can also use gel filtration technique to remove any altered product of interest from the product stream. Gel filtration is a commonly used chromatographic technique that can remove certain impurities from the product stream. It basically works on the principle of size exclusion, where the molecules they can be separated on the basis of their size or shape. Now, one of the basic impurities that can be removed via cell gel filtration is the aggregated form of the aggregated form of the protein of interest. Because the aggregated form has a greater molecular weight when compared to the normal form or the native form, it can be separated using simple gel filtration. Also, an other impurity that can be removed from gel filtration is the extensively proteolized protein. As we know, proteolysis can lead to a formation of smaller protein fragments, which can be separated from the protein of interest using gel filtration. Also, we can remove glycoprotein variants which have which have degraded carbohydrate moieties using gel filtration as well as ion chromatography. Now, different kind of protein modifications such as the deamidation or the oxidation or the incorrect disulfide bond formation in the proteins can also alter the surface charge and shape of the proteins. This can in turn affect the biological activity and stability of the final product. Now, in this case, the chromatographic techniques such as ion exchange chromatography or hydrophobic interaction chromatography are used for the removal of such products from their to remove such products from the other active protein of interest. Now, here you can see different techniques that are used to characterize the protein-based biopharmaceuticals and to detect any protein-based impurities as well. The commonly one being used, the non-naturing gel electrophoresis, the SDS gel electrophoresis, peptide mapping is also important, HPLC, isoelectric focusing, mass spectrometry, amino acid analysis, the N-terminal sequencing, as well as the bioassays and immunological assays. Now, talking about product potency, any biopharmaceutical product, it needs to obey certain product potency specifications. The product potency is usually expressed as the units of activity per vial or per dose or per milligrams. Now, in order to make sure that the biopharmaceutical meets its potency specifications, various assays are performed, including bioassays and immunoassays. Now, bioassays involve the measurement of biological activity of the product, uh, like its ability to stimulate cell growth or inhibit the growth of bacteria. Immunoassays, on the other hand, will involve the amount of product present by using antibodies which are specific to that product. Now, bioassays and immunoassays both have their own disadvantages and advantages, and bioassays themselves are more directly related to the product activity, whereas the immunoassays are more sensitive and specific, but they may not necessarily affect the product's actual biological activity. Okay, talking about bioassays. Bioassays are done by assessing the actual biological activity of the biopharmaceutical. What happens is that we apply a known quantity of the substance or biopharmaceutical and introduce it in a biological system and see how that system responds to that stimulus. That response is then measured, and is that measurement is basically given a quantity to the substance that is being assayed. Now, these bioassays are obviously always comparative in nature because we need a standard preparation against which the sample can be compared. The internationally accepted standards for the preparation of most biopharmaceutical are usually organizations such as the World Health Organization or the USP or United States Pharmacopoeia. Some examples of bioassays is one of those is the antibiotic potency that is measured by the zonus inhibition around antibiotic disc, which is placed on any agar plate that is inoculated with microbes. Another example is the growth factors, which are usually radiolabeled with nucleotide precursors and then incubated with specific animal cell lines, and then check the accelerated growth of cells. Now, the level of radioactivity that would be incorporated into the DNA of the cells is then measured as a bioactivity of the growth factor. So, bioassays for the modern biopharmaceuticals are more complex. They can actually involve whole animals for the bioassay. We could use specific organs or different tissue types or even individual mammalian cells in a culture. Talking about the drawbacks of bioassays, the major drawback is the lack of precision. Due, this is due to the complex nature of the biological system. Now, what happens is that this complexity it can lead to a variability in the responses, which are influenced by factors. It could be the metabolic status of the individual cells or any subclinical infections in the animals. Even the stress that is induced by the human handling, it could also impact the actual results. Now, these factors they can make it difficult to obtain the precise and reproducible results, therefore making it a challenge to ensure consistent product potency. Now, also in addition to the lack of precision and the time constraints, another significant drawback is the cost involved. As we know, bioassays they can be very costly, they can be expensive due to the fact that we are using whole animals, especially during the purchase of the animals, therefore making the routine testing during the downstream processing impractical, and the cost also limits the number of assays which can be performed. Now, due to the previously discussed problems in the bioassays, we are now the modern times are now going with immunoassays. Now, immunoassays are commonly used to detect and quantify the biopharmaceutical products. These basically involve the use of monoclonal or polyclonal antibody preparations that can specifically interact with the product of interest. Now, this basically ensures that the assay is precise. It basically ensures the good assay precision. And now, immunoassays are far rapid and cost effective and straightforward when compared with bioassays. Now, there are two different types of immunoassays that are being used. One is called as RIA, which is the radioimmunoassay, or the EIA, that is the enzyme immunoassay. Now, in the RIA, basically the radioactive immunoassay, the radioactive material is the source, which is used as a marker to detect the antigen-antibody interaction. While in EIA, as the name suggests, enzymes are used as the markers. Now, the markers are used to detect the interaction between the antigen and the antibody, which gives a very reliable and precise measurement of the product. Now, coming towards the ELISA, or the commonly known as enzyme-linked immunosorbent assay, it is an immunological assay, as the name suggests, and utilizes enzymes and markers to detect and specify any antigen-antibody interaction. Now, this technique is very commonly used in research and diagnostic applications. This is because it is a very sensitive and specific technique, and also because of its ease of use. Now, in basically what happens, as you can see in ELISA, The target antigen is first immobilized on a solid surface, usually a microplate, as the ones you can see in the lab as a well plate. Then any specific antibody is added to the surface which can bind to the antigen, forming an antigen-antibody complex. Now, after the formation of the antigen-antibody complex, we wash away any unbound antibody. Then an enzyme conjugated secondary antibody is added. Now this secondary antibody binds to the primary antibody, and the enzyme which is also coming with the secondary antibody, it catalyzes a reaction which produces usually a color with the substrate solution, and this gives a visible signal which is proportional to the amount of antigen that is present in the sample. Now, ELISA can be used to detect a variety of substances. This could be proteins, different peptides, even hormones or drugs, sometimes even DNA. As we discussed previously also, that it can be rapid, and it can be very cost effective and a very straightforward technique. Therefore, which is why the use of immunology in biomedical research has been very much incorporated. Now, despite the clear advantages of immunoassays, there are still certain disadvantages. Now, immunological activity cannot be guaranteed to correlate directly with the biological activity, because even the minor modifications that can occur in a protein, they might not affect its ability to bind to the antibody, but can in actuality reduce the actual biological activity of the product. Immunoassays may provide a convenient, but performing the bioassay and the very least final product is still necessary to prove that the product is within the required specifications. In summary, the biopharmaceutical products involve a series of steps that is required to ensure the safety and efficacy and their compliance with the regulatory standards. Now, these products are complex and can contain impurities, which is why chromatographic techniques are used to separate and purify the proteins of interest to remove these impurities, and the potency of the product is determined through the bioassays or immunoassays, with the latter being the more rapid, cost-effective, and straightforward one. And with this, we have reached the end of our lecture, the lecture of product analysis in biopharmaceutical processes. If you have any questions or any comments and suggestions, please feel free to email me and the given address. Thank you so much for your patience and your time.